Now let me explain the basic mechanism of smooth muscle contraction. First of all, keep it in your mind that important factor for smooth muscle contraction is cytosolic cytosolic calcium, not extracellular calcium, not endoplasmic reticulum calcium. This cytosolic calcium is very important for contraction of smooth muscle. So now we have different ways to enter the calcium to the smooth muscle. First, we will bring and enter calcium to the cytosol, and then we will talk about calcium that have calcium acts and produce contraction in smooth muscle. Okay, now we have different pathways to stimulate smooth muscle. It means we have different pathways to enter calcium to the cytosol of the smooth muscle. We can stimulate a smooth muscle by neurons, okay, specifically by autonomic nervous system or sympathetic and parasympathetic nervous system. For example, we can stimulate a smooth muscle by acetylcholine, by norepinephrine, and so on, and their neurotransmitters. We can stimulate a smooth muscle by different hormones, for example, by oxytocin, by ADH, or vasopressin, and so on. We'll talk about each of them one by one, don't worry. We can stimulate a smooth muscle by some local factors, for example, by nitric oxide, prostaglandin, histamines, and so on. And also, we can stimulate a smooth muscle by stretching it. When we stretch to smooth muscle, so a lot of calcium will be entered to the cytosol of smooth muscles. And also, sometimes smooth muscle generates automatic action potential or depolarization and repolarization, which are called slow waves and spike potential. Okay, now let's talk about each of them one by one. First, we can enter calcium to the smooth muscle by production of action potential or by production of depolarization and repolarization. How? Let's see it. First of all, in a smooth muscle, resting membrane potential is around negative 60 millivolts. Okay, and threshold potential in smooth muscle is around negative 40 millivolts. Okay, and in skeletal muscle, resting membrane potential is around negative 90 millivolts. Here, because we have a lot of leaky channels for cations and some other factors that we will talk about them, all of them work together and they cause the intracellular of smooth muscle to be less negative or to be in negative 60 millivolts. Now this is threshold potential. This is a point, this negative 40 millivolt in smooth muscle. When we touch it, when we bring our smooth muscle to negative 40 millivolts, so depolarization and repolarization in smooth muscle will be started. This is a very important point. Now, sometimes spontaneously, without any external stimulation, the smooth muscle reaches to its threshold potential. Okay, and these waves are called slow waves. Now let me tell you that what is the basic mechanism of slow wave production. For example, first of all, its mechanism is not so much clear, but anyway, there is some uh, hypothetical ideas related to slow wave production. For example, we have a pump which is called sodium potassium ATPase or sodium potassium pump, and its function is to pump 3 sodiums from the inside of the cell to the outside of the cell, and 2 potassiums from the outside of the cell to the inside of the cell. Now, sometimes this pump works slowly. When it works slowly, for example, when it works slowly, so it cannot pump more positive charges or more cations from the inside of the cell to the outside of the cell. And when you cannot pump a lot of positive charges, so positive charges build up inside the cell, and these positive charges bring our intracellular voltage from negative 60 to negative 40 millivolts. This is one point. Okay, now another point for this is that we have some channels here which are called the leaky channels. For example, this channel is for calcium, and we know that we have more concentration gradient of calcium extracellularly. So calcium flows from high concentration gradient to low concentration gradient, and calcium is a cation. And also we have some, for example, sodium leaky channels, and sodium is also a cation. So when we add more cations, more positive charges to the inside of the cell, they also bring our intracellular voltage from negative 60 to negative 40 millivolts. Okay, now when we reach a peak point of slow wave, okay, automatically this slow wave will be dropped back, and this dropping of slow wave is due to fast working of sodium potassium pumps. When this pump works so fastly, so it will pump more positive charges, and the slow wave comes back to its basic level or to its normal range. Okay, plus when we reach to the peak of slow wave, okay, due to these more calcium, for example, when we reach to this negative 50 millivolts, and this point some potassium channel will be opened, and these potassium channels are related to calcium. Okay, sometimes they're called BK channel or big potassium channels. So now more potassium efflux occur, and it also causes the slow wave to reach and come back to its uh, basic or to its normal range. Okay, now this was the mechanism of slow wave production. Now if I touch threshold potential, it means if I reach to negative 40 millivolts, so automatically a lot of voltage gated calcium channel will be open. For example, these are caveoli, and in caveoli we have a lot of calcium voltage gated channels. Now when we reach to the peak of this slow wave, or when we reach or when we touch negative 40 millivolt, okay, due to this opening of calcium voltage gated channel, a lot of calcium enters to the inside of the cell. Okay, and a fast depolarization is produced, and at the peak of depolarization, once again potassium voltage gated channel will be opened, and a lot of potassium leaves the cell. Okay, and our cell once again reaches to its normal range, which is called repolarization. So this production of this wave is called spike potential. Okay, so spike potential is produced by the help of slow waves, and keep it in your mind that one slow wave can produce many spike potentials. And now we know that by the help of spike potential, by the help of slow waves, we entered a lot of calcium to the cytosol. So this is one way to enter calcium to the cytosol, and we'll talk about the calcium later on, that what does calcium do with our cell laterally. Okay, so this is one way of entering of calcium. Now next way to enter the calcium, next way is also so easy. When we stretch a smooth muscle, for example in our gut lumen or in our GI tract, when we eat a lot of food, so when we stretch these smooth muscles, here we have some channels which are called mechanical gated channels. They will be opened, and a lot of positive charges enter to the inside of the cell, and also a lot of calcium enter to the inside of the cell, and these positive charges bring our resting membrane potential to a threshold potential, and in threshold potential once again spike potentials or depolarization and repolarization will be produced, and this is the next way that we can enter calcium to the smooth muscle. This is plateau phase. We will talk about plateau phase in cardiac muscle. Okay, and for example this first part of this type of wave action potential is due to calcium entry. This next part is, which is called plateau phase, due to potassium efflux and calcium influx, and this last part is due to potassium efflux. We will talk about it and cardiac muscle. So till now we have studied two ways of calcium entering to the cytosol or smooth muscle. Now let me talk about third way of calcium entry to the smooth muscle, and this way is very important because we have some receptors on smooth muscle. We have different types of receptors for different ligands. Okay, receptors are proteinous structure on the cell membrane. Okay, that when we stimulate a receptor, they cause production of nerve events inside a cell, so they are called receptors. And anything which stimulates receptor, this is called ligand. Okay, now for example this is acetylcholine. Okay, acetylcholine here is a ligand. This acetylcholine is a ligand. Now we have a type of receptor on smooth muscle for acetylcholine, which is 7 pass receptor. Okay, and we have already studied about 7 pass receptors in previous lectures. Now for example this is a 7 pass receptor. We have different, don't get confused, we have different types of 7 pass receptors. For example we have alpha 1 type of 7 pass receptor, we have alpha 2, we have beta 1, beta 2, beta 3, we have muscarinic type 1, 2, 3, 4, 5. All of them are types of 7 pass receptors, and we have different ligands for different receptors, and each of them works in different ways. Got it? For example this is a type of receptor which is for acetylcholine, or this is a muscarinic receptor. Okay, now when we stimulate this muscarinic receptor by acetylcholine, so this receptor will activate a beautiful protein inside the cell which is called G protein. Now, like 7 pass receptor, we have different types of G proteins also. For example, we have GS protein, we have GI protein, we have GQ protein. But here my example is about GQ protein. This is a globular protein. And this 7 pass receptor is also called G protein coupled receptor because this protein is coupled with a globular protein inside of the cell. Now, you for example you stimulate this cell by this receptor by acetylcholine. Now this 7 pass receptor will be activated. Now due to stimulation of this 7 pass receptor, G protein or GQ protein will be activated, And we have three domains, three subdomains of G protein, which is called G. For example, this is called alpha Q domain. This one is called beta Q domain, and this one is called gamma Q domain. Now, this alpha Q domain, after stimulation of 7-pass receptor by acetylcholine, this alpha Q domain is disassociated. This will be disassociated. Okay. Now, this alpha Q domain activates a beautiful enzyme which is called phospholipase C. And phospholipase C enzyme is located in inner leaflet of the cell membrane. Now, this phospholipase C enzyme degrades a beautiful type of phospholipid which is called phosphatidyl N-acetyl diphosphate, or PIF2. Now, this phosphatidyl N-acetyl diphosphate is dissociated into two basic parts. One part is called phosphatidyl inositol. This part is called diacylglycerol, and this part is called phosphatidyl inositol triphosphate. Now, phosphatidyl inositol triphosphate, or IP3, comes here, and here is, this is sarcoplasmic reticulum, and we have a lot of calcium inside the sarcoplasmic reticulum. Now, inositol triphosphate activates or stimulates a receptor, a channel-like receptor, which is called IP3 receptor. It means this receptor is for inositol phosphatidyl inositol triphosphate. Now, when we stimulate this receptor by IP3, so the gate will be opened, and we have a lot of calcium inside the endoplasmic reticulum, and a lot of calcium flow from the inside of the endoplasmic reticulum to cytosol. And now we have a lot of calcium in cytosol once again. So this is the third pathway that we can increase cytosolic calcium by this pathway. So we talked about depolarization and repolarization in smooth muscle. We talked about mechanical gates or stretch pathway in smooth muscle. We talked about calcium entry by ligands or by stimulation or receptors. Okay, and here is another way also for calcium entry. For example, when this calcium, which is inside of endoplasmic reticulum, when its level decreases, okay, this endoplasmic reticulum calcium, so here we have a beautiful sensor to this calcium. This sensor is called stromal STIM1 protein. Okay, and for the first time this was discovered in stromal cells. Now, this STIM1 protein is a sensor for calcium. When this protein detects that inside of the endoplasmic reticulum the calcium level is going down, okay, so it will activate automatically a type of calcium channel which is called ORA channels, or they are called store operated calcium channels. It means these channels are depending on the store of calcium inside the endoplasmic reticulum, and when you open this channel, so a lot of calcium enters to the inside of the cytosol. And also we can use this calcium for two purposes. One, we can use this calcium for mechanism of contraction. Another, we can use this calcium to fuel the store of endoplasmic reticulum by this, because this calcium is literally pumped to the endoplasmic reticulum by a beautiful pump which is called circa, that we'll talk about it later on. So up to now, we did a beautiful thing. We entered a lot of calcium to the cytosol through different ways. And keep it in your mind that simultaneously a smooth muscle can be stimulated only by one way, or by one neurotransmitter, or by one local factor, or it can be stimulated by different factors in same time. Got it? Now let me talk about this calcium. The calcium which is present in cytosol, and we have it in cytosol now. Now, this part is so easy. This calcium which we have entered them, okay, this calcium comes and it will join to a beautiful protein inside the cytosol which is called calmodulin. This is called calmodulin. Now, and calcium plus calmodulin, both of them are going to make a beautiful complex, and this complex is called, this complex of calmodulin plus calcium, this complex is called calcium calmodulin complex. This is called calcium calmodulin complex. Now, calcium calmodulin complex is going to activate a beautiful enzyme which is called myosin light chain kinase. It means kinase are this group of proteins, okay, that they are going to phosphorylate, they are going to add a phosphate on other proteins, and phosphate is energy. And all those proteins which are going to add phosphate on other proteins, all of them are called as a group kinase proteins. And myosin light chain means that we have here myosin, okay, and in previous lecture we studied about myosin, that myosin is made of three basic parts. It's made of a head part, it's made of a neck part, and it's made of a tail part, okay. And this is actin or thin filament. Now, this is the rule when you want to contract a smooth muscle or a skeletal muscle or a cardiac muscle. We'll talk about it here. This is the rule that you have to connect head of myosin to the thin filaments, and due to this connection between these two, contraction will be produced. That we'll talk about it here, don't worry. Now, in this neck part, in each of these myosin molecule, we have two proteins, and they are called this one is called essential light chain, and this green one is called regulatory light chain. Now, myosin light chain kinase means that this protein adds phosphate to these light chains of myosin, specifically on regulatory light chain. These tails and heads are heavy chains, and these proteins are light chains because they're smaller and wet. When you add phosphate to regulatory light chain, or when you add phosphate to light chains of myosin, it means you activated myosin. And now myosin is ready to produce contraction. We'll talk about it here, don't worry. Now, this myosin light chain kinase has two important functions. One, it adds phosphate on regulatory light chain kinase and activates these myosins. Secondly, before contraction, these myosins or thick filament are separated from thin filament or actins by two important proteins and deactivated by the two important proteins. And these two proteins are called calponin and caldismon. Calponin and caldismon. Now, this myosin light chain kinase also inhibit and deactivate calponin and caldismon. And now this thin filament is absolutely free to join to thick filament. Plus, third function of myosin light chain kinase is that it activates ATPase enzyme of myosin. And we know when we do contraction process, we need energy or ATP, and we have to change ATP to ADP. Before myosin light chain kinase activity, this myosin or this thick filament cannot change ATP to ADP. But when we add phosphate to it, it's now active. It can change ATP to ADP, and it's free now to join to thin filaments. And now let's watch its mechanism of contraction, that how this thick filament and thin filament together produce contraction. This is so easy. Listen me carefully. This state, this is a very beautiful state. Here, this, for example, step 1. And step 1, these thick filaments, these are thick filaments here, these are thick filaments, these are thin filaments, okay? And now we want to join these two together, and we want to bring these actins to this area, to the middle port, like a side polar mechanism. And we want to bring these two dense bodies together, and we want to produce contraction. Okay, now in this step number 1, myosin and actin is inhibited by troponin, not by troponin, by calponin and by caldesmin. Now, in this step 1, we have ATP. Already we have it. Okay. Now, when we add a phosphate to this myosin by myosin light chain kinase, okay, we activated ATPase enzyme of thick filament. So now thick filament is ready to convert ATP to ADP plus phosphate. So now we converted ATP to ADP plus phosphate. This is phosphate and this one is ADP. Plus, we removed and deactivated this calponin and caldesmin also. So this is stage number 2. And stage number 2, when we hydrolyze ATP to ADP plus phosphate, so this head of myosin will change its position. It will cock back, okay? And this is now ready to join to this thin filament in step number 2. And this cocking back of the head of myosin is due to a hydrolyzation of ATP into ADP plus phosphate. And step number 3, the head of myosin will be binded to the active sites of actin automatically. And in step number 4, in this step, okay, and it means from step number 3 to step number 4, between these two steps, we will lose a phosphate molecule, and we have only ADP. And this attachment will be stronger furthermore, okay, due to losing of phosphate. And in this step, it means from step 4 to step 5, okay, we have already ADP with us plus a head of myosin will change its position from back position to front position, and it will bring the thin filament from this area to this area, okay? And now cross bridge occurred. Now in this state, from step 5 to step 6, we will lose ADP also. So now in step 6, we it means from step 6 to step 1, we will add ATP. When we add ATP, automatically this head of myosin will be detached. Okay, and now we're once again in previous stage. Now once again ATP will be, now for example, now this in this state, this head of myosin, this is thin filament. Now I'm in this state, so I'll hydrolyze ATP into ADP in phosphate and head cocked back to next to new position. And this state, when in this state, when I lose phosphate, I'll join automatically to new position. Okay, and when I lose ADP plus phosphate, also both of them, so I'll bring this thin filament from back position to anterior part. And now in step 6, I'm in this position. When ATP comes and it binds to myosin, so myosin head will be detached and it's ready for next cycle. And that's why we bring this. For example, these were the head of mycins. We brought this thin filament from this area to this area, and we brought this thin filament from this area to this area. It means we brought these two dense bodies toward each other, and we produced contraction. Okay, so this was cross bridge cycle and ATP hydrolyzation and ATP usage. Okay, now this is the last part. Don't worry, this is easy. Now at the end of contraction, be careful here. We don't have this protein in skeletal muscle. Here, when you want to terminate contraction, when you want to terminate this recycling, you need an enzyme here. Okay, and this enzyme is called myosin light chain phosphatase. Okay, and activity of this enzyme is depending on calcium or cytosolic calcium, because at the end of contraction we will pump all calcium from the cytosol to the sarcoplasmic reticulum by a pump which is called SERCA. Okay, that it pumps hydrogen from the proton from the inside of into plasmic reticulum to cytosol, and it pumps calcium from the cytosol to the endoplasmic reticulum, and it uses ADP or energy. Plus, we will pump some calciums from the cytosol to the extracellular area by a beautiful pump which is called sodium calcium exchanger. Okay, so we will pump a lot of calcium also from this area to the outside of the cell. Plus, at the end of depolarization, some potassium channels will be opened, or some potassium voltage gated channel will be opened, and a lot of potassium ions leave the cell, and once again cell becomes in its normal state or in its resting state. And now we don't have calcium here. So when we don't have calcium here, when we don't have positive ions here, this myosin light chain kinase will be deactivated automatically because we don't have calcium. So this will be deactivated. When this myosin light chain kinase is deactivated, so this will not work. Plus this myosin will not work. Plus an enzyme will be activated which is called myosin light chain phosphatase. Now its function is, myosin light chain phosphatase function is to remove a phosphate, to remove phosphate from this myosin. And when we remove phosphate from myosin, so it means we deactivated myosin once again. And now this myosin will work slow. Okay, the cross bridge cycle will be slowed. Okay, and finally this contraction mechanism will be terminated. Okay, because when you remove phosphate from the myosin, it means now you cannot break ATP to ADP. Okay, and also calponin and caldesmon are once again coming to their original position. Okay, So this enzyme is very important. So this was the end of smooth muscle contraction or relaxation state. Okay, now come here. This is so easy. In start of the lecture I said that we have two types of smooth muscles. One type is called multi-unit smooth muscle. Another type is called unitary smooth muscle. Multi-unit smooth muscle means that they are stimulated one by one individually and they contract one by one individually. That's why they are called multi-unit. And it means different units contract individually one by one. But here, this bundle of smooth muscles are connected to each other by gap junctions. When you stimulate one smooth muscle, it will stimulate its neighbor smooth muscle. It will stimulate its neighbor smooth muscle, and they will stimulate it each other automatically. So they will contract all of them together. That's why they are called unitary smooth muscle. It means they contract as a one unit. Now, this type of muscle, and also these small uh, parts. This is a neuron. Okay, these small parts of neurons are called varicosities. And instead of axonal bulb, we have varicosities here. Okay, now these multi-unit smooth muscles are located in some parts of our body. For example, here, this is our skin. Okay, this is our skin. This is our hair follicle. Okay, the hair on our skin. Now, this hair follicle is connected to our skin by a smooth muscle. This is a smooth muscle, and this is a multi-unit smooth muscle. And this smooth muscle is called erector pili muscle. When you stimulate this smooth muscle will be contracted, and it causes this hair to stand up, to be straight, and causes standing of our hairs on our skin. This is one type of multi-unit smooth muscle. And other parts of our body, for example, this is our eye, and we know about eye. I'm not going so deep inside the eye, but anyway, this outer layer of the eye is called sclera. This middle red ear is called choroid, and innermost layer is called retina. Okay, this front bulging part of our eye is called cornea. Okay, cornea, and this part is called iris, which gives color to our eye. And this part behind the iris is called lens. This is called lens. And function of lens is accommodation phenomenon. It means it makes our vision far and near. Okay, don't worry. Now here we have a muscle. Okay, this muscle is called ciliary muscle. This muscle is called ciliary muscle. Ciliary muscle. And this ciliary muscle is connected to the lens by these small black ligaments, and they are called zonular ligaments. Zonular ligaments. Now, when we contract this ciliary muscle, for example, when we contract this ciliary muscle, so these zonular ligaments become loose. Okay, now when they become loose, okay, this lens is going to be flat, or its wide is going to be more, and this causes near vision. Okay, so now this is not important for me. For me, this is important, that this ciliary muscle is made of multi-unit smooth muscles. Okay, you don't need to learn about the lens, about the iris, about any part of the eye. We'll talk about them inshallah in eye anatomy. Okay, don't worry. And other places for This multi-unit smooth muscle, for example, in our body is the iris. Okay, for example, this is pupil. This black point in our eye is called pupil. Now, around the pupil, we have these multi-unit smooth muscles in a circular fashion. When they contract, okay, they causes constriction of pupil. And also we have some muscles around the pupil, okay, or in iris, that when they contract, they causes dilation of pupillae. Okay, so in a nutshell, we can find multi-unit smooth muscle in different parts of our body, like rector pili, like ciliary body, like lens. Okay, because in some books they discussed this things over these examples, so that's why I brought them here. We talked a lot about the smooth muscle. We talked about these mechanisms, about we talked about slow wave mechanism, we talked about stretch mechanism in smooth muscle. We talked about ligands, local factors, and also action potential, and also relaxation of smooth muscle. Keep it in your mind, don't worry about receptors. When you study physiology, okay, you will learn about each type of neurotransmitter one by one. You will learn about norepinephrine, that how norepinephrine stimulates our smooth muscle, how acetylcholine stimulates our smooth muscle, how histamine stimulates our smooth muscle. This was only one example for you that we can stimulate a smooth muscle by ligand also. Okay, now these tiny terms. Latch state mechanism. Latch means lock state. Okay, it's very important. Okay, once the head of myosin binds to thin filament, it will not be detached automatically and sooner. Okay, this is very important. They will stay in lock state. They will stay for a moment in a lock position. Okay, and this locking state is called latch state mechanism, because latch means locking. Okay, now what is mechanism of latch state? Two important things. First, when we want to detach head of myosin from actin filaments, we need myosin light chain phosphatase enzyme. Okay, and myosin light chain phosphatase enzyme doesn't work so faster. Okay, so it needs a little bit time, okay, to put a phosphate on a myosin and to detach them from each other. So for a moment, for a millisecond, they will be in attached state. Okay, plus ATP cycle in smooth muscle is slower than the skeletal muscle. When you want to convert ATP to ADP, or when you want to add ATP to thick filaments, so this cycle is a bit slower than the skeletal muscle. So that's why when we produce action potential, or when we produce contraction in smooth muscle, so thick filament and thin filament will be in lock state for a moment. And it has a benefit. It helps us to use less ATP, okay, and produce more force. So this is economically very good, and also it produces a tonic contraction. For example, in our blood vessels, we need this type of mechanism to produce long contraction and maintain our blood pressure. Okay, so this is the benefit of latch state mechanism. Another term here in smooth muscle that we don't have it in skeletal muscle, it means that contraction in smooth muscle is slow, and also relaxation of smooth muscle is also slow, related to the skeletal muscle. And this is called slowness of onset and relaxation of smooth muscle. And this also because of two mechanisms. First, in smooth muscle, we have latch state mechanism, and latch state mechanism needs a little bit time. Okay, and also here calcium entry, here ATPase activity, all of them are slow related to the skeletal muscle. So that's why they will contract slowly. Calcium enters to the smooth muscle slowly. That's why they contract slowly. And also we have elasticity mechanism, so they will relax also slowly. So both relaxation and contraction needs a little bit time in smooth muscle. For example, in smooth muscle, a contraction cycle is occurred around 2 up to 3 2 up to 3 seconds. Okay, but in skeletal muscle this contraction is occurred around 0.05 seconds. So this is slow contraction. Here is slow, then the smooth, then the skeletal muscle. Another important thing is stress relaxation and reverse stress relaxation. It's so easy. If if you stretch a smooth muscle beyond its normal, it will not lose its force, it will not lose its tension compared to the skeletal muscle. In skeletal muscle we studied, if you increase the length of the skeletal muscle, so it will lose its force. Okay, but here when you stretch smooth muscle, it will not lose its force, it will maintain its force once again. And this is due to high elasticity, more elastic factors, and arrangement of these thin and thick filaments in this smooth muscle. And also if you decrease the length of the smooth muscle, and this is called reverse stress relaxation, if you decrease the length of the smooth muscle, okay, so once again they will maintain their tension and pressure and their normal range. Okay, and this is also due to elasticity. And we have an example for this in our bladder. When you put a lot of urine in bladder, okay, so it will be stretched but it will not lose its force or its tension. But when you decrease the size of urinary bladder, okay, so it will not lose its force, it becomes once again normal. Got it? So these are all about smooth muscle contraction, smooth muscle histology, and about these mechanisms and differences between smooth muscle and skeletal muscle. So please, if you haven't subscribed to our channel or liked our channel or shared our channel, share it, subscribe it, and like it. Have a good time till next lecture.